शीतला सस्यश्यामला मातरम् वन्दे मातरम् शुभ्रज्योत्स्ना पुलकितयामिनी पुल्लकुसुमित द्रुमदलशोभिनी सुहासिनी सुमधुरभाषिणी सुखदां वरदां मातरम् वन्दे मातरम् वन्दे मातरम्.
Okay, good morning students. In the last class we learned about cell organelles. So in the cell organelles today we will going to learn about some more cell organelles. Some of the important cell organelles today also we will going to learn. Okay, so in that the first one, mitochondria. What is that? Mitochondria. Mitochondria is one of the important cell organelle in the cell. Mitochondria are the one of the important cell organelle in the cell. If you see the structure of mitochondria, so look at the board. This is mitochondria. This is mitochondria. So if you observe mitochondria, there are two layers in the mitochondria. That is called outer membrane. This is called outer membrane, and second layer is called inner membrane. So in the mitochondria, how many layers are there? Two membranes are there, outer membrane and inner membrane. So next, actually there are two chambers also is there, two chambers. This is called, this empty space is called outer chamber. What is that? Outer chamber. So inside the layer there is a chamber like structure, that is called inner chamber. So actually this inner chamber is also called as matrix. This inner chamber is also called as matrix. So if you observe inner membrane, there are some fold-like structures. These folding-like structures are called cristae. What is that? Cristae. Actually, what is the shape of this mitochondria? That is spherical, cylindrical or circle. So many shapes are there. The mainly spherical or cylindrical. Okay, if you see this length, long, what is the length of this mitochondria? 2 to 8 microns. What is the length? 2 to 8 microns. If you see the width means vedalpu. So wide means that is 0.2 microns. Length, that is 2 to 8 microns. If it is wide means vedalpu, 0.5 microns. So actually in one cell nearly 100 to 150 mitochondria are there. Nearly, if you see the structure of the cell, inside the cell, nearly 100 to 150 mitochondria are there. Okay, so already I told what is cristae, what is mitochondria. Is it right? Inner foldings are called cristae. So inside the inner membrane there is empty space. That space is called matrix. Okay now. So actually, what is the main purpose of this mitochondria in the cell? What is the main purpose of this mitochondria in the cell? The main purpose of this mitochondria in the cell, that is cellular respiration. What is the use of this mitochondria? For doing cellular respiration. Inside the cell, the respiration process, most of the respiratory process takes place in the mitochondria. So when the mitochondria doing the cellular respiration, meanwhile a little amount of energy is released. The total energy is stored in the mitochondria, in the form of ATP. Already I told you, no? So actually the total energy is stored in mitochondria in the form of ATP. What is that? ATP, adenosine triphosphate. What is that? Adenosine triphosphate. So actually because of this reason, this mitochondria is having energy. So that's the reason these mitochondria are called as powerhouse of the cell. What is that? Powerhouse of the cell. కణశక్తి భాండాగారము ఇన్ తెలుగు వాట్ వి సే కణశక్తి భాండాగారం అంటే కణములో శక్తిని కలిగి ఉండేటువంటి భాగం, ఎక్కువ భాగం శక్తిని కలిగి ఉండే భాగం ఏంటంటే ఏ ఆర్గనేల్, సెల్ ఆర్గనేల్ అంటే అది మైటోకాండ్రియా. సో దట్స్ ద రీజన్ ద సెల్ ఈజ్ కాల్డ్ యాజ్ the mitochondria is called as powerhouse of the cell. Next one, endoplasmic reticulum. What is that? Endoplasmic reticulum. So endoplasmic reticulum, this is also one of the important cell organelle in the cell. If you see the structure of the cell, inside the cell there is a network like structure. There is a network like structure. This network like structure is called endoplasmic reticulum. This network like structure is located in the cytoplasm of the cell. What is that? Cytoplasm of the cell. Actually this network like structure, it creates some of the passages. Passages means like గొట్టాలు లాంటి నిర్మాణాలు, passages. So this network like structure, it creates or it's form a passage like structure. So with the help of these passages, some of the important substances transport from one place to another place in the cell of the cytoplasm. For example, the wanted substances passing through this channel from one place to another place in the cytoplasm of the cell. One place to another place. How it is traveling, the substances in the cytoplasm of the cell? Through this channel. What is this channel? It is a network like structure. This network like structure is called endoplasmic reticulum. What is that? Endoplasmic reticulum. It is useful for transport some of the substances in the cytoplasm of the cell. Okay. Actually, if you see the structure of the endoplasmic reticulum, for example, this is endoplasmic reticulum. This is called upper layer, this is called lower layer. So if you see clearly, if you observe, actually if you want to see this structure of this endoplasmic reticulum, that is not possible through our naked eye. It is, if you want to see this endoplasmic reticulum in the cytoplasm of the cell, that is possible to see through only electron microscope. So when we observe this with the help of electron microscope, we can find out the structure of this endoplasmic reticulum. If you observe this endoplasmic reticulum, on the top layer there are some granule-like structures are there. On the top of the layer, some of the granule-like structures are there. So because of this granule-like structures, if you touch this endoplasmic reticulum, it look like a rough, మీన్స్ అంటే గరుకుగా ఉన్నట్టు కనిపిస్తుంది అనమాట. అంటే ఆ ఛానల్లో పైన ఉండేటువంటి పొర అంతా కూడా గరుకుగా ఉండేలా కనపడుతుంది. సో ఈ గరుకుగా ఉండేటువంటి ఈ లేయర్ని ఏమంటారంటే రఫ్ ఎండోప్లాజ్మిక్ రెటిక్యులం. రఫ్ ఎండోప్లాజ్మిక్ రెటిక్యులం. So the downward portion, if you observe, it is very smooth. It is very smooth. So actually this down endoplasmic reticulum is also called as smooth endoplasmic reticulum. Why? Because there is no granule-like structures. There is no granule-like structures in the downward membrane. Is it right? So according to this nature, the endoplasmic reticulum, two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is having granule-like structures. They are called ribosomes. What is that? Ribosomes. So this ribosomes, what is the use of these ribosomes? These ribosomes, it synthesize proteins. These ribosomes, it synthesize proteins. What is the smooth endoplasmic reticulum? What is the use of this smooth endoplasmic reticulum? It prepares lipids. It will prepare lipids. So that's the reason this endoplasmic reticulum is having proteins and lipids. These proteins and lipids are transport in the cell to different parts where they need by this endoplasmic reticulum. The proteins and lipids are manufactured and transport to various places in the cytoplasm of the cell through this endoplasmic reticulum. That is the main use of this endoplasmic reticulum. Not only that, some of the biochemical activities also, will do this in this endoplasmic reticulum. This is the main purpose. So some of the vertebrates, వెన్నెముక గల జీవులు, some of the vertebrates, it plays a key role. Means it is a crucial role. What they are doing in the vertebrates? In the vertebrates, deoxifying poisonous and drugs. Means వాటిని ఏం చేస్తారన్నమాట? నాశనం చేస్తారు, వినాశనం చేస్తాయి అన్నమాట. ఏమన్నా ఫాయిల్ అంటే విషపూరితమైన పదార్థాలు ఏమన్నా ఉంటాయి కనుక వాటిని ఏం చేస్తాయి? డియాక్సిఫైయింగ్. చూడండి, డియాక్సిఫైయింగ్, విడగొట్టేస్తాయి, నాశనం చేస్తాయి. దట్ ఈస్ ద మెయిన్ పర్పస్ ఇన్ vertebrates with the help of this endoplasmic reticulum. This endoplasmic reticulum, it's spreading throughout the cell. Where it is? In the cytoplasm. It is a channel-like structures. It's having two layers. First layer, it is rough in the nature. The second layer, smooth in the nature. According to that, the endoplasmic reticulum, two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is having ribosomes. The ribosomes are useful for preparing or synthesis proteins. Smooth endoplasmic reticulum is preparing lipids. So that's the reason this endoplasmic reticulum transport the proteins and lipids in the cell where they need. So in vertebrates, it is very plays a crucial role, that is deoxifying poisonous drugs and poisonous drugs. Okay, next organelle, that is Golgi operators. What is that? Golgi body or Golgi operators. Golgi operators or Golgi bodies is also one of the cell organelle in the cell. Who discovered this Golgi body or Golgi operators? Camillo Golgi, the scientist name is called Camillo Golgi. First he find out this Golgi operators in the cell. So that's the reason that organelle is called as Golgi operators or Golgi bodies. In the year of 1898. Actually, if you see the structure of this Golgi operators, it's made up with several layers. So the end portion of the layer look like a sac-like structure. The end of this layers, it's formed as a sac-like structures, means sanchilanti nirmanalu. From these sac-like structures, a vesicles are formed. Vesicles means gundrati nirmanalu. See, vesicles. These are called vesicles. This is the structure of this Golgi operators. Sac-like structures, vesicles. Actually, what is the main function of this Golgi operators? Already we saw endoplasmic reticulum. On the top layer of the endoplasmic reticulum, ribosomes are there. And the down layer, that is called smooth endoplasmic reticulum. From the rough endoplasmic reticulum, proteins are produced. And from the smooth endoplasmic reticulum, lipids are produced. So, these proteins and lipids are entered into this Golgi operators. In the endoplasmic reticulum, the proteins and lipids are produced. From that, these are entered into these Golgi operators. So, in the Golgi operators, there are some vesicles. It is entered into these vesicles. The proteins and lipids are entered into these vesicles. Here, when this proteins and lipids and some other substances are entered into this Golgi operators, in that all the materials are manufactured, means packed. From this, after manufacture, all the substances are entered into lysosomes. From the transport means where it is transport? It is transported to the lysosomes. Lysosomes also one of the cell organelle in the cell. So, from the Golgi operators, the substances are transported to the lysosomes. From the lysosomes, all the materials are reaches to the plasma membrane. For example, this is cell. It reaches to the plasma membrane. From this onwards, the substances are utilized. If it is waste materials, it is sent out. If it is useful things, used by this plasma membrane. Sometimes these substances are useful for participated in the repairing of the plasma membrane. If it is unwanted substance, it is sent out by this membrane. Okay, na? So, likewise, the substances are transported from this. Here, the food material, the substances are packed and transport to the lysosomes. From the lysosomes, it transport to the plasma membrane. Sometimes these substances are participated in repairing of the plasma membrane. Sometimes the plasma membrane it may get damaged. So during that time, the substances are participated in the repairing of this plasma membrane also. So actually, these Golgi operators in all the cells, not in the same quantity or same number. Cell to cell, the number of Golgi operators changed. In secreting organs means just like salivary glands, liver cells, mammary glands. They all are secreted some of the chemicals. Is it right? The glands are secreted some of the chemicals. For example, if it is salivary glands, saliva secreted by the salivary glands. So in the secretory glands, this Golgi operators number is more. Why? Because in the secretion process, these Golgi operators are participated. If it is mammary glands, these Golgi operators are participated in the secretion of the milk. If it is salivary gland, these Golgi operators are participated in the more secretion of saliva. So, in cell to cell, the number of Golgi operators are changed. So, the number of Golgi operators is more in secretory glands. Where there is secretion process takes place, in that cell, the number of Golgi operators are more. Okay, next. Okay, next. The next organelle, that is called lysosomes. What is that? Lysosomes. For scientists, they didn't understand. In the cell, there are some destructive enzymes. Means కణాన్ని నాశనం చేసేటువంటి enzymes they found in the cell. సో కణాన్ని నాశనం చేసే సెల్స్ కణంలో ఉంటే కణం ఏమవ్వాలి? డిస్ట్రాయ్ అవ్వాలి. బట్ ద సెల్ డిడ్ నాట్ డిస్ట్రాయ్. దే ఆర్ గెటింగ్ కన్ఫ్యూజన్. అరే, ఈవెన్ దో there are some destructive enzymes in the cell, but the cell did not get destroyed. What is the reason? They didn't understand. But later they found that there are some tiny particles. They found in the cytoplasm. The tiny particles in the cytoplasm are called lysosomes. When they absorb these tiny particles in the cytoplasm, these tiny particles is contained in the tiny particles. Means in the lysosomes, they found the destructive enzymes. It means these destructive enzymes not in the even though it is in the cell, but in the cell where it is there, it is in the lysosomes. Because of that, the cell will not get damaged. Means all the destructive enzymes are in the lysosomes. What is the main use of this lysosome? But if there is no lysosomes, these destructive enzymes are freely in the cytoplasm of the cell, then the cell will be damaged. But all the destructive enzymes is in the lysosomes. So that's the reason the cell will not get damaged. What is the main function of these lysosomes? When the unwanted substances, when the waste materials, when the poisonous substances are in the cell, these substances are sent into these lysosomes. Then already inside the lysosome some destructive enzymes. With the help of these destructive enzymes, these materials are destructed. నాశనం చేయబడతాయి అన్నమాట. ఎక్కడ? లైసోజోమ్లో. ఎవరి చేత? ఈ డిస్ట్రక్టివ్ ఎంజైమ్ చేత అవి నాశనం చేయబడతాయి అన్నమాట. So after destruction in the lysosomes, these lysosomes, it's nearly come to the plasma membrane, then it burst. అవి పగిలిపోతాయి అన్నమాట. పగిలిపోయినప్పుడు అందులో ఉండే పదార్థాలు సెల్లోకి వచ్చి ఆ సెల్లోకి వచ్చిన తర్వాత అవి డైజెస్ట్ అయిపోతాయి. ఇట్ మీన్స్ ఇవి డిస్ట్రక్టివ్ ఎంజైమ్స్ని ఏం చేస్తాయి? తనలో అవి నాశనం చేసి ఆ డిస్ట్రక్టివ్ ఎంజైమ్ చేత తర్వాత వాటిని బయటికి పంపించేస్తాయి అన్నమాట. అవి బయటికి పంపించాలంటే ఆ యొక్క లైసోజోమ్ అనేది ఏమవ్వాలి, పగిలిపోవాలి. సో వెన్ ద లైసోజోమ్స్ ఆర్ బర్స్ట్, దీస్ ఆల్ ద మెటీరియల్స్ ఆర్ రిలీజింగ్ ఇన్ టు ది సెల్. సో ద సెల్ విల్ బి డైజెస్టెడ్. అది ముందు డైజెస్ట్ చేసుకోలేదు, అది సెల్ ముందు డైజెస్ట్ చేసుకోవడానికే అవకాశం లేదు, ఎందుకంటే అవి విషపూరితమైన పదార్థాలు అన్నమాట. సో ఆ విషపూరితమైన పదార్థాలు ఈ లైసోజోమ్స్లోకి వెళ్ళి ఆ లైసోజోమ్లో అవి నాశనం చేసిన తర్వాత వీటిని మళ్ళీ బయటికి పంపేస్తుంది ఎవరు, ఈ లైసోజోమ్. దెన్ దిస్ లైసోజోమ్స్ బర్స్ట్, దెన్ ద మెటీరియల్స్ ఆర్ రిలీజింగ్ ఇన్ టు ద సెల్. ద సెల్ విల్ బి డైజెస్టెడ్. సో ఇవి తనంతట తానే పగిలిపోతాయి అన్నమాట సెల్లో అవసరం వచ్చినప్పుడు. ఆ పదార్థాలు బయటికి పంపించడం కోసం అవి పగిలిపోతాయి అన్నమాట తనంతట తానే. ఒక విధంగా చెప్పండి, దాన్ని ఏమంటారు, అది సూసైడ్ చేసుకుంది అంటారు అన్నమాట. ఆత్మహత్య చేసుకుంది. ఏం చేసుకుంది, ఆత్మహత్య చేసుకుంది అనమాట. సో దట్స్ ద రీజన్ ద లైసోజోమ్స్ ఆర్ కాల్డ్ యాజ్ సూసైడల్ బ్యాగ్స్ ఆఫ్ ద సెల్. వాట్ వీ కాల్ సూసైడల్ బ్యాగ్స్ ఆఫ్ ద సెల్. ఆత్మహత్య కోశాలు అంటాం తెలుగులో. అర్థమవుతుందో చెప్పింది ఏంటి, ఆత్మహత్య కోశాలు. ఇట్స్ ఎ టైనీ పార్టికల్ ఇన్ ద సైటోప్లాజం ఆఫ్ ద సెల్. చాలా చిన్న చిన్న రేణువులు వంటి నిర్మాణాలు ఈ లైసోజోమ్స్, కానీ పెద్ద పని చేస్తాయి అనమాట. వీటి యొక్క ఫంక్షన్ ఫ్రమ్ ద బిగినింగ్ దే డింట్ అండర్స్టాండ్ టు ద సైంటిస్ట్స్. శాస్త్రవేత్తలకు ముందు అర్థం కాల. ఏమని అర్థం కాల, చాలా రకాల డిస్ట్రక్టివ్ ఎంజైమ్స్ అంటే వినాశనకరమైన ఎంజైమ్లు సెల్లో ఉన్నప్పటికీ సెల్ ఎందుకు డ్యామేజ్ అవ్వట్లేదు వాళ్ళకి అర్థం కాల. దెన్ దే కేమ్ టు ద నో when they find out the lysosomes. Next, one of the important cell organelle which is present in the plant cell only, not in animal cell. One of the important cell organelle which is present in the plant cell only, not in animal cell. That is called chloroplast. What is that? Chloroplast. It is one type of plastid. What is plastid? Plastid mean రంగులను కలిగి ఉండేటువంటి నిర్మాణాలు. వాటిని మనం ఏమంటాం, ప్లాస్టిడ్స్ అంటాం. ప్లాస్టిడ్స్ అంటే రంగుల్ని ఇస్తాయి అన్నమాట. ఏమిస్తాయండి, రంగుల్ని ఇస్తాయి, ప్లాస్టిడ్స్. సో chloroplast is also called as one of the plastid which is present in the plant cell only, not in animal cell. Up to now what we discussed that all the organelles are present in the both plant and animal cells, but this plastids means this chloroplast is only present in the plant cell, not in animal cell. Means in the animal cell the plastids are absent, chloroplast is absent. Okay, here if you observe the plastids are two types. Chromoplast and leucoplast. Chromoplast అంటే ఏంటంటే రంగులు ఇచ్చేటువంటి ప్లాస్టిడ్స్. మరి ల్యూకోప్లాస్ట్ అంటే అవి రంగులు ఉండవు, కలర్లు ఉండవు, కలర్లెస్ ప్లాస్టిడ్స్ అనమాట. ల్యూకోప్లాస్ట్ అంటే వైట్ ఇన్ కలర్, మేబీ కలర్ వేరే రంగుల్లో ఉండవు ఆ ప్లాస్టిడ్స్ అనేవి. ఇవి డిఫరెంట్ కలర్స్లో ఉండే ప్లాస్టిడ్స్ అనమాట. So what we observed that plastid that is called chloroplast, that is green in color. The plastid green in color. So that's the reason it is come under leucoplast. Sorry, come under chromoplast, not leucoplast. Come under chromoplast. Okay, what is the shape of this chloroplast? Disc shape, oval shape. Some of the plants may be ladder in shape or star shape. No, different shapes. If you find out, these chloroplasts are having different shapes. Next, what is the diameter? Means, what is the diameter? The diameter of this chloroplast 4 to 10 microns. అది ఎంత మందము ఎంత వెడల్పు ఉంటుందంటే 4 to 10 microns the diameter. So what is the main function of this chloroplast in the plant cell? What is the main function of this chloroplast? The main function of this chloroplast to trap the solar energy and convert it into a chemical energy. to trap the solar energy and convert it as a chemical energy. Do you know this process is called? This process is known as photosynthesis. What is that? Photosynthesis. In the process of photosynthesis, this chloroplast to trap the sunlight or to trap the solar energy to turn into a chemical energy. This process is known as photosynthesis. So because of this reason, the plants are preparing their own food. The plants are preparing their own food. Okay na? So if there is no chloroplast, that living organisms, they can't do the process of photosynthesis. Why plants are doing photosynthesis? Because of this chloroplast. When the chloroplast, see some of the plants, they may not do the process of photosynthesis. Why? Because in their plant body, these chloroplasts are absent. Some of the plants, not all the plants, some of the plants. But one who is having chloroplast in their body, they can do the process of photosynthesis and preparing their own food. They are called autotrophs. This is about chloroplast.